Founded in 1998, Bioshaf Limited develops, manufactures, and markets innovative systems for the medical diagnostics market. Bioshaf's first product is a complete system for fertility diagnostics. The system is comprised of sets of kits covering the most common causes of infertility, 25 parameters in all, for both the male and female, a flow cytometer, and proprietary software that runs the tests and provides a complete printout of the results within 2 hours. Bioshaf Limited also developed a line of diagnostic kits for a variety of clinical uses. Here you can see a demonstration of the semen analysis kit. First, the semen sample is diluted by a buffer and divided to a set of 7 tubes. A for control, B for motility, C for viability, D for counting white blood cells and the number of immature sperm cells, E for anti-sperm antibodies, F is for the morphology of sperm's head, and G is for sperm cell count. For tubes B to G, 10 microliters of a specific reagent is added. Reagent B to tube B, reagent C to tube C, and so forth. The tubes are then incubated for 30 minutes at room temperature, and washing buffer is added to all except tube G. After centrifugation, the pellet in each tube is resuspended with buffer and is ready to be read by the flow cytometer. With Bioshaf's unique program loaded, a reading frame opens. The flow cytometer sensors then analyzes 10,000 cells from each tube, and the data is collected and processed. As soon as the last tube in the set is analyzed, the software program displays the final results. A biological function called acronym reaction is critical for the fertilization ability of sperm cells. In this kit, the normality of this function is tested. Sperm cells are washed and resuspended to the original volume of the sample, and 0.2 microliter are then transferred to tubes A, B, and C. Then several reagents are added to tube C. This mixture of reagents is able to rapidly induce the acronym reaction. The cells are then incubated at 37 degrees Celsius for 30 minutes. When the incubation time is ended, a monoclonal antibody, specific to antigen exposed only after the acronym reaction occurred, is added to the cells in tube C, as well as to the sperm cells in tube B as a control. After an additional 30 minutes incubation at room temperature, the tubes are analyzed by the flow cytometer. As soon as tube C is analyzed, the software calculates the percentage of sperm cells that underwent the acronym reaction as compared to the control tubes to give the final result and printout report. This third kit detects the level of autoantibodies to thyroid's antigen in sera samples. To the first 6 tubes, differing levels of thyroglobulin autoantibodies are added, and this set of 6 tubes is used to draw a calibration curve. Positive and negative control serum are added to the next 2 tubes, followed by the test samples. In the next step, beads coupled with thyroglobulin antigen are mixed with the serum samples. After a short incubation time, a second fluorescent-labeled antibody specific to human immunoglobulins is added to detect the specific thyroglobulin autoantibodies. The reading process is simple, and the result of the tested sera are calculated from the calibration curve formula.